Hello friends, welcome to a new topic on cytogenetics, that is role of hypotonic potassium chloride in karyotype. The role of hypotonic potassium chloride during harvesting in karyotyping is mainly used for uh, this hypotonic potassium chloride helps in cell swelling, and due to cell swelling the chromosome spreading will be better, which is necessary for to get a good morphology of chromosome. Now let's see the mechanism of action. So the hypotonic solution that we used in uh, cytogenetic laboratory is 0.075 molar. So this hypotonic solution, it means it has a lower solute concentration than inside the cell. Due to the difference in solute concentration, water rushes into the cells by osmosis in an attempt to equalize the concentration. The influx of water causes the cells to swell significantly. The swelling helps to separate the tightly packed metaphase chromosomes. So let's understand here. So this is three jar number 1, 2 and 3. The first jar, it has the medium which is hypertonic. The second jar, it has the medium which is hypotonic. And the third jar, it has the medium which is isotonic. And we have placed a cell inside the medium. Here one cell, here one cell, here one cell. So here the cell is placed inside a hypertonic medium. Here the cell is placed inside a hypotonic medium, and here the cell is placed inside a isotonic medium. Now let's understand what is mean by hypertonic. Hypertonic means it has the medium has the solute concentration more in comparison to the solute concentration inside the cell. So if the solute concentration outside of the cell is more, it means the solvent concentration will be less in comparison to the solvent concentration inside the cell. So as there is a concentration difference between inside the cell and outside the cell, to make it balance, the solvent will or the water will come out of the cell to make it balance. Okay, so here in hypertonic solution, the water will come out of the cell through the process of osmosis. So the cell will shrink. Here the cell will shrink in a hypertonic medium. Now in hypotonic medium, the solute concentration is less in comparison to the solute concentration uh, inside the cell. So here if solute concentration is less, it means solvent concentration will be higher in comparison to the solvent concentration inside the cell. So to make the balance, the solvent or the water from outside of the cell, it rushes into the cell to make the balance, so that the cell, due to influx of water, the cell will swell, or the cell will increase in size. The cell will swell here. Okay, so in hypotonic medium the cell will swell. Now let's come to the isotonic medium. Here the solute concentration outside of the cell and the solute concentration inside of the cell is same. It means solvent concentration also same. So there is no any difference between the concentration. That's why there is no any movement of water, or there is no any movement of solvent. So the cell will remain as it is. So in hypertonic solution, the cell will the cell will shrink. SH. Here the cell will swell, and here the cell will remain as it is. Okay. Now let's see the results in karyotyping. What we'll get by using this hypotonic potassium chloride? We will get better spread of chromosome, because due to treatment of hypotonic potassium chloride, the cell will swell and it will become fragile, which helps in nucleus to burst when we drop the pellet from a height, and we'll get the individual chromosome, because due to swelling of cells, the chromosomes, they are well spread, and when they burst onto the glass slide, we'll get a better spread metaphase chromosomes. So this treatment, hypotonic potassium chloride treatment, is crucial for high quality metaphase spread, where individual chromosomes do not overlap, allowing proper counting, pairing, and bending analysis necessary for karyotyping. Now, in summary, the hypotonic potassium chloride treatment is a vital step in cytogenetic sample preparation, as it leverages the principles of osmosis to enlarge the cell and disperse the chromosomes, directly leading to well-separated metaphase spreads required for accurate karyotyping analysis. So, due to hypotonic potassium chloride, uh, we through the process of osmosis, we get the enlarged cell, which helps in well separated metaphase spreads, which is necessary for accurate karyotypic analysis. So this is the use of hypotonic potassium chloride in cytogenetics. If you like the video, please like, share, and subscribe to the channel uh, to get more upcoming videos on cytogenetics. Thank you.